All right, welcome everyone, and I'm just going to open it up to any questions that you have to begin with. I will share my screen and show you whatever is relevant to your questions. Let's see, I do have our study guide up on the screen now. So, does anyone have any questions to start off with? Okay, Siobhan, why don't you start? Mm-hmm. Okay. So this is the so the, in the, this we're talking about the innate immune system, right? So we have certain inborn features of our anatomy and physiology that are protective against infection, and um, and so one of them is just the physical barriers like the skin and the mucous membranes, but there are features of those physical barriers that are chemical. So, for example, in our skin, we have the oils that are secreted from our follicles that have antimicrobial properties. So they have short fatty acids that are inhibitory to the growth of bacteria. Also, our skin happens to be a low pH, so acid, like you say, um, which also is inhibitory to a lot of types of bacteria, not the ones that are normal microbiota on our skin, but others. And then it's rare that uh, a pathogen can cross that physical barrier unless they have some help, like a vector of like a mosquito that could just pass the pathogen directly to the blood across that barrier of the skin. So the other thing is that in our mucous membranes, we also have chemicals. And in our mouth, for example, we're always putting food and fingers and pens and things in our mouth, right? But we're not getting sick all the time. So why is that? We have our saliva, right, which has enzymes like lysozyme that will help to limit the growth and the viability of any pathogens that are present in our food or whatever we put in our mouth. Um, and the mucous membranes themselves also have enzymes like lysozyme that's produced. So those are the chemicals. Like they're really the molecules, the enzymes, and the acidity of the skin, the acidity of the gastric juice in our stomach. That's what I mean when I'm referring to that. Okay? Yeah. Yep. Yep. Right. Mhm. Yep. Another question? Aaron or Ulrich, you guys have? Okay, so yeah, this is chapter 17. Really, I'm focusing on two sections in chapter 17. I believe it's 17.4. And artificial immunity, by definition, is something that your body doesn't produce, right? So it's artificial. It's something from outside your body. But it's generating immunity, i.e., protection from infection, from disease, really. The pathogen can still enter the host, so it can still infect, but it won't be able to cause disease, right? Um, and that's the goal, is protection of a vaccine, for example. We could also get artificial immunity from an injection of, say, antibodies like immunoglobulin that would temporarily protect and boost our immune system and help us to fight off an infection. So there are two forms. There's um, passive, which is temporary, and active, which is lifelong, can be lifelong if you get all the booster shots associated with certain vaccines. Um, and this is from sections 17.6, talking about vaccines, and table 17.5. So in that table, it mentions a lot of vaccines, but what I wanted you to pick out about polio and what I went over in my lecture was about the two forms of polio vaccine. There's an attenuated and inactivated. Oh, here. Okay. Share screen. Okay, so let me show you from the notes of chapter 17. Oh, is that what you were referring to? Okay. So, not the, it's not, it can be the route of vaccination, but in the case of polio, they're the two forms, right? So what's the difference between these two forms is in this slide here. Let me make it bigger so you can actually see. So the the polio vaccines are named differently. They're Salk, which and Sabine, named after the people who discovered them and developed them. And they took two different routes about the same time in history. And the inactivated one, by definition, this is just kind of a classic example of what the difference between the two main types of vaccines are. We also have other types of vaccines, but these are the key for most of the vaccines that you get as a child. They'll either be inactivated or attenuated. So what are the benefits and risks associated with these different forms? Well, um, in the attempt to eradicate polio as a disease, um, they're both very effective because by definition your vaccine has to have as close to 99% efficacy as possible to protect everyone. Otherwise, people who get vaccine may not have that lifelong immunity that we were talking about, the artificial immunity. Um, and so then they could, if exposed to the pathogen, to the virus in this case, they could get sick from it even after the vaccine. But both of these vaccines are very effective, more than 90% effective, but they have different features. So inactivated means that the virus is dead. It can't, even if it gets in a susceptible host, it cannot replicate. Even if it got in a host cell, it could not replicate. Okay? And that's usually because it's been chemically kind of fixed in time. But the chemicals like formaldehyde that were used in this case, it's not always formaldehyde, but it can be, um, it kind of freezes the structure of the virus so that all of those surface antigens that would elicit an adaptive immune response are there and preserved as much intact and in their natural form as possible. And that's the idea, whether you have the inactivated or the attenuated, you have to present the pathogens, you have to present the antigens that are specific to that pathogen in order to get immunity. If for whatever reason those antigens and the epitopes associated with them are not preserved, you won't get lasting immunity. So there are different ways that you can do that. When it's inactivated, it has to be injected in order to elicit that immunity. And that's in the case of polio vaccine and any inactivated. Like the flu vaccine, we have an inactivated form. And that's when you get the flu shot. But we also have the flu mist. That's an attenuated vaccine. In this case, the attenuated vaccine is a drink. And that's because the polio virus, the way that we get sick from it, is that we ingest it the virus in contaminated water. Used to be that maybe a little over a century ago, that people, because there was poor sanitation, people would always be exposed during their childhood to polio virus. And they would get some protection when they were very young from, if they were nursing, from their mother's milk, because she would also be exposed and would have antibodies circulating that would be passed through the milk to the child and protect them. So that's one way of passive natural immunity. It's like a temporary immunity that we get from during infancy from our mother. But this vaccines that are artificial immunity that are active, i.e. creating the memory cells, creating the antibody generating cells that will be able to protect you if you get exposed later in life. That's only by vaccination or by actually getting polio. But the risk with polio is that you can get poliomyelitis, which is the paralytic form of polio. And so it's better if you can get the immunity by the vaccine, right? Because the vaccine, by definition of vaccine, the benefit has to outweigh the risk. So it has to be something that elicits this immunity, lifelong immunity, but doesn't give you the disease, right? So the key differences between these two forms are, I kind of line them up side by side. One is injected. That means it's more expensive to administer because it has to have a trained medical professional to administer the vaccine. Okay, so when we're thinking about worldwide vaccination, that's a challenge. The inactivated form requires booster shots, so you have to follow up with those in the population that have already gotten one. It has good immunity, good protection, lifelong, once you get that booster. But it's safer because you're not getting into your body a live pathogen that's able to replicate. In the other case, the attenuated, it's the entire virus still alive, able to replicate if it gets in your host cell, into the cells of the epithelial cells lining the intestines in this case. It's weakened by passaging it usually in another species, so in this case monkeys, and then putting it back in the human as a vaccine. It's easy because you just drink it, and it doesn't require any refrigeration, which an inactivated vaccine was, so it's easier to administer. That was when the Rotary Club started this effort to vaccinate the world and eradicate the world of polio. It chose the oral vaccine because it was just easier and cheaper. They didn't have to hire medical professionals. They could get volunteers. And it only requires one dose. There's no follow-up. One dose gives you long-term immunity because it basically mimics the actual infection cycle, only it's weakened enough so that you're protected from getting the paralytic poliomyelitis. Unfortunately, it's in very rare cases you can get paralytic poliomyelitis from the vaccine. Most of the cases in which that occurred, it was because somebody made a mistake in the production of the vaccine, and they were getting not a weakened virus but a more virulent virus. But even with the properly produced weakened virus as a vaccine, it still in very, very rare cases can cause paralytic poliomyelitis. So, and it gives a different kind of immunity because instead of being injected right into the blood where you're not getting exposed to the mucous membranes, you're getting it down into, you're taking it, ingesting it, it's getting down into your uh, intestines, where that's where the virus is going to infect its native host cell, right? And cause an immune response and cause IgA immunity. So any time that you get the actual virulent form of the polio, that you're exposed to it and it gets in your body, then the IgA will immediately be there to protect you from getting sick, right? So it'll IgA, remember, this is some of the stuff that you have to remember about antibodies or immunoglobulins, is that they protect from viruses by neutralizing the virus before it's able to enter the host cell, right? They bind up the outside, all of those antigens on the outside, and so those antigens can't, the spikes that are present there on the outside, they can't interact with the receptor on the host cell. And without that interaction, they'll never get into the host cell and able to replicate. So that's the idea behind either IgG or IgA, but it's more relevant here for IgA because if you have just IgG immunity, the virus can get into to your gut, and then once it gets into the blood is when the IgG is going to have a protective effect. So it can still, the virulent form of the virus can still get into your host cells in the gut. Okay, so the other advantage, especially for worldwide vaccination, is that the shedding, so this is the fecal oral route, right, of poliovirus. So if you actually get the virulent form of the virus, you're getting it through contaminated water due to unsanitary conditions, right? That in that same way, you can get the vaccinated weakened form only in the case of the Sabin or the attenuated form of the vaccine. That's not true of the inactivated. You won't shed anything because it's not alive, so it won't replicate enough. You'll shed a little bit of the vaccine when you get it, but it's not enough to have an effect on exposing those around you to the virus. And once they get exposed, yeah, it wouldn't be enough. If they got exposed by the oral route, it wouldn't elicit any immunity. It has to be injected through the blood, right? So that's a big difference as well. And the safety issue is an important one because even here in the U.S., when I was a kid, I got the oral vaccine for polio, and you also got a vaccine for polio. Does anyone remember which form you got? Well, you guys got so you might, Ulrich, you might have gotten oral. Where did you get vaccinated when you were a child? Okay, so yes, oral. But for Americans, it's going to be like my kids. They got the Salk. Yeah, it's actually combined. It's called the DTP. So there's diphtheria, tetanus toxin, and so diphtheria, tetanus, and oh no, that's pertussis. Sorry, my mistake. Yeah, but it's going to be injected for the polio vaccine. So it's going to be a shot. We don't get it orally anymore. And actually the reason that is because we have so few cases of polio that when you looked at the risk and the benefit in the U.S., the risk was greater of getting polio from the vaccine rather than from some live virus that was circulating in the population. And so all of a sudden the choice of vaccine was clear that, okay, we can't use the attenuated anymore because there's this risk of actually giving it to a child and actually causing them to have paralytic poliomyelitis, which is suddenly greater than the risk of them getting it naturally. So that safety is a big difference between these two forms of the vaccine. Sorry, that was a long-winded answer, but it's it's important to understand these differences, all of them. Um, but the safety issue is a big one in deciding what's the benefits and what's the risk. All right. Any other questions? Yeah. Right. Yeah. Yeah, there's a figure. Let's see. I think I wrote it down. Yeah, in chapter 18, they go over toxins, right? And let me just open those notes. So in the chapter 18 notes, I think I have a figure directly from the book, but it has all of these 9 modes of action of because they're we already know from labs like the hemolysins, they lyse the membrane, the target of within the host cell is a membrane target. But there are other modes of actions. There are neurotoxins. There are that. Actually, I think we had somebody present in class about the botulinum neurotoxin and showing us what those, um, how the synapse, the signaling across the synapse is blocked there. Oh, I don't have it in my notes. Oh, I just skipped over it, huh? 'Cause I have it here. Oh yeah, here are the toxins. Right. Yeah, this is it. So this is, I think, figure 18.23 in your book. Let me get down. So it shows these. I think the only one missing here is the neurotoxin, which is at the synapse specifically, not in any cell. It's in a neuron. So it would be things like the neurotoxin from Clostridium botulinum, things like that. But the others, yeah, plasma membrane can be disrupted, the cytoskeleton can be changed, and that's part of the pathogenesis of many bacteria is that they alter the shape of the cell that they're trying to attach to. Remember, in pathogenesis, there are key steps. Uh, the pathogen has to enter the host, has to attach and colonize, then it has to evade the immune response long enough, not forever, but long enough for it to kind of grow and colonize and maybe switch on the set of genes it needs, those virulence factors it needs to cause disease. It may be producing a toxin and having those effects. And then it will uh, what else? Oh yeah, cause the damage, right? And then it will exit and try to find a new, uh, oops, there's some, are you guys hearing that background noise? Let me just, okay. Uh, I, I think I can modify it. I don't know who the, oh, call-in user, here we go. Okay, is that better? Sorry, is that better? Can you hear now? Okay. All right. Yeah, so there are various, so the cytoskeletal alterations are actually, I think we had a presentation on that as well. Um, this, where the often gram-negative bacteria will use this secretion system called the type 3 secretion system to directly insert the toxin in the host cell that they're trying to attach to and colonize, and it will change the structure of the membrane. So, for example, Shigella um, can get engulfed, can induce a host cell within the gut to engulf it and into an endosome by uh, by delivering these factors or toxins inside the host cell and causing the membrane to ruffle around it and then bring it into the cell. And then E. coli also have this system where they will modify the cytoskeletal structure to make these pedestals that they kind of sit on top of, and it means that they have more access to nutrients than the other bacteria that are just flush against the surface of the tissue at that site. So protein synthesis can be stopped. We have diphtheria toxin from Clostridium diphtheria as an example of that. And then the cell division or the cell cycle steps can kill the host cell, cause it to die as a result. And yeah, signal transduction. So some of these are specific to a certain cell type. Um, Signal transduction is this step where a receptor, something on the outside of the cell that is able to be detected by the host cell, and then inside the cell will change its behavior. It will turn on genes as a result, or it will open channels as a result. The cholera toxin opens the sodium in the channels, and then as a result of the exit of sodium, we get an exit of chloride and water that causes that watery watery diarrhea that's so characteristic of cholera. Um, uh, in the case of Staphylococcus, it has this toxin called exfoliative toxin that loosens the connections between the cells of your skin, and as a result you get this scalded skin syndrome that kind of looks like your whole top layer, the epidermis, has been kind of removed and it's all red and raw, and then you're susceptible to secondary infections as a result. And then the vesicle trafficking, um, so movement of things within the cell and or out of the cell in the case of exocytosis, which is important for the neurotoxins like Clostridium botulinum, like preventing that signaling at the synapse. So those are all the ways, all the various ways that and you only really know, you don't need to know the examples, you just need to know kind of where in the cell the targets are, whether it's affecting the cytoskeleton or the plasma membrane or the nucleus in the case of cell cycle, those kinds of things. Any other questions? Go back to, so you guys all set on these essay topics. The evasion of the innate immune response is the open response question that we did for homework, asking about how bacterial pathogens like Shigella and Salmonella, and I think it was Coxiella was an example as well, avoid phagocytosis. or can survive the process of phagocytosis. So for something like that, you need to review what are the phagocytic cell or the phagocytes, excuse me. What is the process of phagocytosis? How does it kill bacteria? And then how do these pathogens, in particular Shigella and Salmonella, avoid, the avoidance of the immune system? How do they avoid that, the end result of phagocytosis? And then genetic changes, we've also gone over in an open response. You can look back to, I think this goes back to chapter 12 actually, because there's not a lot of virus examples in chapter 18. But this is the genetic changes that account for um, either the seasonal or the pandemic. So there are two changes, right? And I think we had also a presentation on this, so it should be familiar. But there's a genetic shift, which is a large change that would result in most people not having immunity. And so it could be pandemic, could spread quickly in a population because influenza is very readily spread, easily spread by that respiratory route with any kind of droplets, whether you're talking or sneezing or coughing, and also because it can survive on fomites surfaces. And then the next person who touches that surface might then introduce the virus into their body. And then the seasonal differences, which are smaller, so we call them genetic drift. They're smaller point mutations that would account for small changes, enough of a change in the surface antigens on the virus such that our immune system wouldn't have a strong immunity from the previous season. And so we would still get sick, still get symptoms if we were introduced to the strain of the virus that's circulating the next flu season. And then the role of the exotoxins, we went over some of the different kinds of toxins. Remember that cholera toxin is the one I'm talking about is the one that disrupts the signal transduction. And it uses this receptor to enter the cell with its the B subunit, which is the binding part of it. Do I have that in here? Yeah, here it is, the two subunits. So the A subunit is the toxic, the B is the part that binds the cell. Once it gets in, it breaks apart through cleavage, proteolytic cleavage, and the A part exits into the cytosol, and that's where it has its effect on the opening the channels. So the chloride is, it influences cyclic AMP, and if there's too much cyclic AMP, it causes the secretion of chloride ions, which leads to also sodium and water loss from the cell, and cell death from some of the cells. So you get this kind of, they call it rice water stools, where it's very watery and it has little white flecks in it. They're actually the dead cells from your intestines. And then finally the last topic, the artificial immunity, we already went over with the polio vaccine. So those, if you know those, then those topics really well, then you'll be pretty much set for the rest of the questions on the exam. And I did just want to mention, because I know someone in class had asked about what we have to know about the microbes that I've named here on the second part of the study guide. And so I just want to spend a little bit of time going over that. So the important thing here is that you remember what they are. So as far as what group they belong to in the microbial world. Are they bacteria? Are they archaea? Are they fungi? Are they yeast? Which would fungi would be a group of that, but single-celled fungi are yeast. And are they protozoa or non-living members of the microbial world like viruses, right? So if I give you polio without telling you it's poliovirus, you should know that it's a virus that causes polio. Or influenza, you should know it's a virus, it's not bacteria. I've pretty much put them in order of the first group up to from Clostridium up to Mycobacterium tuberculosis. Those are all bacteria, different species of bacteria. And then the ones marked viruses are all viruses. Plasmodium has different species. Plasmodium malaria is one of them, but Plasmodium falciparum is the most virulent form that causes malaria. It's a protozoa. So also Candida and Cryptococcus are both fungi, but Candida is yeast. It's a single-celled fungi, and Cryptococcus grows as mycelia, kind of in this branching kind of like like the mold in your refrigerator, kind of on a piece of old food or something. It's kind of that fuzzy growth that you see. That's characterized by branching, kind of root-like growth. So other than the group, you should review why these groups are different, such as their cell structure, which ones have a prokaryotic cell structure, which ones have a eukaryotic cell structure, and which ones don't have a cell structure. They're acellular. And then, so I'll just list a few features that I've made notes about for myself here. So starting with Clostridium, I'm not going to ask you about what disease they cause. I'll probably give that information if it's necessary, but you should know, for example, Clostridium are gram-positive and they are endospore forming. E. coli are gram-negative. They have this type 3 secretion system that they have attachment factors like pili. They have antigens that help to distinguish the different strains within the pathogenic species. The difference between the pathogenic type of E. coli and the normal microbiota non-pathogenic forms are that the pathogens like enterohemorrhagic E. coli produce toxins, whereas our normal microbiota do not, so they can't harm us in that way. Remember that because E. coli is gram-negative, it has the endotoxin, but it also, in the pathogenic strains, produces exotoxins, so these potent toxins that are in a protein form. Vibrio cholera, also gram-negative, so it has endotoxin, and the pathogenic, again, there are Vibrio cholera strains that are non-pathogenic, and there are strains like L. tor or the classical pathogenic strain that produce exotoxins like the cholera toxin. Staphylococcus aureus, we know it has many more toxins and it has many exotoxins, but it's gram-positive. So the ones that I'm going to ask you to remember what gram status they are these you don't have to worry about about Streptococcus, but certainly E. coli and Staphylococcus aureus you should know from lab that which one is gram-positive, which one is gram-negative. But Staphylococcus aureus the pathogenic strains, again, we do have this as normal microbiota. It's not a toxin-producing strain in that case, but the pathogenic forms that are toxin-producing include the production of things like toxic shock syndrome toxin, but it has other toxins as well that let's go back to that because I'm forgetting what the names of them are. So, like hemolysins, leukocidin, which will, the alpha toxin is the hemolysin, which will lyse red blood cells. The leukocidin is the leukocyte or white blood cell specific toxin that will lyse those white blood cells. So it has efficacy against both red blood cells and white blood cells. Let's see. Shigella, I'm more, like I said, for the open response question, I'm more concerned with your knowledge about how it escapes from the phagosome, but it is a toxin-producing pathogen as well. Salmonella also can escape from the phagosome. So key virulence factors that are associated with these bacteria that you would have read about, or key structural features like endospore formation for Clostridium, or in the case of Mycobacterium, it has this waxy cell wall that's the outermost layer, the mycolic acids, that's the outermost layer of the cell. In the case of HIV, influenza, and polio, these are all viruses. With HIV, the relevant part of its virulence is that it's actually infecting a cell that's an important part of our immune response. So the T helper cells are incapacitated when the HIV is actively infecting them and killing them. And then influenza viri, we already talked about genetic shift or drift, which is key for that. Poliovirus, the two forms of the vaccine, which we already talked about. Plasmodium has multiple forms, which makes it hard to control. It also is vector mediated, so remember it's passed from human to human or from animal to human by mosquitoes. And so it has a sexual life cycle in the mosquito, asexual outside of the mosquito, in our own red blood cells and liver cells. And then two forms of fungi. Candida is can switch between the yeast or the unicellular form and the mycelia form of other types of filamentous fungi. And so we call it dimorphic, or two shapes. And then the features of growth of Cryptococcus as far as filamentous growth, mycelia. And these are important, especially in Candida. It can switch to the mycelia form, which can help it invade our tissues once it gets into us. So Candida is actually part of our normal microbiota for the females, and when it becomes imbalanced with the bacteria, which often happens due to changes in diet or changes in your menstrual cycle, you can get candidiasis, or a yeast infection. And that's basically the Candida is able to grow and becomes imbalanced. So it kind of overgrows within our body and causes symptoms as a result, an infection as a result. And Cryptococcus is waterborne, pathogenic form of fungi. Um, the thing about fungi is that they're usually opportunistic pathogens, so Cryptococcus included. Usually have to be sick with something else, or there has to be some kind of weakened immune system as a result of medication or some other disease for you to get sick with fungal pathogens. So those are my notes for um, the specific species that I've listed on the study guide. Any other questions? Yeah, for example, well, you should, for example, these won't, some of these will be relevant, like um, um knowing that Vibrio cholera is a gram-negative is relevant to understanding whether the kind of toxins that it produces, and it actually produces both exotoxins and it has endotoxin. But I think most of you, if you just do a quick review of gram-positive, gram-negative, that should be familiar already. Like I said, reviewing the cell structure, prokaryote, eukaryote, acellular in the case of viruses, these different groups from the microbial world. Um, but uh, things like the innate immune response, so you should be studying that to understand, to gain the knowledge that you need to answer the short essay question. And so questions or topics like describe physical barriers would be under that umbrella already. So that should already be included in your, and the chemical barriers that we talked about that are also part of the innate immune system. Things like the inflammatory response, or the how fever helps us to limit the growth or kill the bacteria in our body. Um, and then I'd say the only other thing you need to know other than, so obviously in this first topic you need to know about phagocytosis. I think I have that down here somewhere. This that you should be able to, I forget where it is, but something about label a diagram. Yeah, here it is. Describe how phagocytes recognize foreign cells, each of the steps of phagocytosis, be able to label a diagram. Complement, you should know at least and that's not covered in these topics up here, you should know at least how complement is activated. We always have it in our blood circulating, but it's in an active form. How is it activated? You may already know this from anatomy and physiology too. Um, and what are the outcomes? So what does it do to a pathogen? Does it tag it? One of the outcomes is it opsonizes, so it tags it with the complement protein so that the phagocyte can recognize it and bind it and start the process of phagocytosis. Another one is it will kill. In the case of gram-negatives, it can actually assemble many complement proteins on the membrane of the gram-negative bacteria, and by forming this tunnel or series of proteins, it basically pokes a hole in the membrane. And if there's a hole in the membrane, then that selective permeability is lost, and the cell will lyse and die. So those kinds of things you should know, the activation and the outcome. And there's just the basics of how does adaptive immunity, which is covered in the vaccine question. So you should know about adaptive immunity. How do vaccines work? They elicit adaptive immunity, right? So that memory cells, where the first time that you would see the pathogen or as a vaccine, the weakened pathogen or the inactive pathogen, um, you're going to get the symptoms. You're going to get an infection that leads to an immune response. In the case of vaccine, you don't get the symptoms with that, but in the case of the actual pathogen, you would get the symptoms and with it. But then the secondary response, right, is what is protecting you in artificial immunity and also just natural immunity, getting sick from it, from the polio. If you were to get exposed and recover, then you wouldn't get it again in your life. You would be immune to it because of that uh, adaptive immunity. The second time that, or the third time, or the fourth time that you're exposed to it, you're getting uh, protection, and that's really clear in the graph that's shown in your, I think it's chapter 16. Um, that you have a whole much larger response as far as the amount of antibody that's made and the time lag between when you get exposed to the pathogen and um, when you make that antibody response. So let's just look at that graph for a moment, because this is really key to our understanding of this. The primary response, you get both IgM and IgG made, but you can see the IgG is concentration is peaks, but it's not as high as the secondary response, right? Also, see where that peak is. It's probably about 5 to 7 days after the antigen exposure, whereas here it's maybe 1 to 2 days, right, in the scale. So you're getting a really fast response and a really dramatic response with tons of IgG being produced by those memory B cells, which are giving you protection, right? So those functions of antibody are really important here, because that's basically your immunity. I wonder if I have the list. Yeah, neutralization, immobilization, depending on what kind of pathogen it is. If it produces toxins, it can neutralize the toxin, can neutralize a virus um, from infecting a cell. Let's see. Can stop motility of bacteria by binding their flagella. It can bind bacteria that are non-motile together, clump it together as far as agglutination, or if it's some kind of soluble factor that's produced and secreted by the bacteria or other pathogen like fungus or protozoa. Opsonization is important, so not only complement proteins will coat a pathogen, but also antibodies will coat a pathogen and start that process of phagocytosis. Complement activation by the classical pathway, in this case, antibody is involved in, and antibody-dependent cellular cytotoxicity. So NK, or natural killer cells, will recognize the other end, or the Fc portion of antibodies that are bound to a pathogen. And this is usually for large eukaryotic pathogens like helminth or even protozoa that are killed in this way if they're multicellular. Let's see, back to the list. Oops. Can't seem to get there. The basic difference between B cells and T cells, B cells being the ones that recognize extracellular pathogens, mostly, for the most part, that's bacteria. T cells recognizing intracellular pathogens, so by definition that's virus, because they need to get into our host cell or they can't replicate. So again, active and passive immunity should be something that you study for this fourth topic for the short essay. Um, One thing that I do want you to understand from all this is the difference between a pathogen and a non-pathogen. So I've talked about like E. coli being normal microbiota, but then we also know that E. coli can be life-threatening pathogens as well, right? So what's the difference? It's a whole host of things, but the main uh, differences can be summarized as it's able to attach and colonize um, even with the presence of the normal microbiota there, which usually will kind of take up the space and use up a lot of, compete for nutrients, etc. So the pathogen has to be able to have some features that help it and maybe give it an advantage for obtaining nutrients. So one of those examples could be like Staphylococcus has the alpha toxin that can lyse the red blood cells and release the iron that it needs in order to grow. Um, it also obviously has to avoid the immune system. So let's say that you've perforated your gut and your normal microbiota E. coli are entering a space that is usually it doesn't occupy, right? So it can make you sick, but your immune response um, will be able to clear that pathogen much more easily than an enterohemorrhagic E. coli, which is producing um, a toxin um, that's then can spread to your kidney and cause kidney failure, right? So that normal microbiota doesn't have that feature, so it won't cause those other kind of symptoms that you get. But yeah, normal microbiota in the wrong place, certainly it can cause an infection. If you get a cut, the bacteria that are normally on your skin can cause redness and swelling and an inflammatory response. But that is going to be localized to the cut, and um, it unless there's some pathogenic bacteria in there, it shouldn't be able to spread to um, the rest of your body, right? It doesn't have that feature where it can spread, where it can avoid phagocytosis like salmonella do, and then get disseminated by staying in the phagocytic cell and going to the lymph node and all those kinds of things. So I think it's not that you can completely ignore what's down below, but use that to be sure that you have all the information you need um, for these essay topics, and then you should be fine. Yeah, should be well prepared. So some of them are redundant, like explain the genetic changes, antigenic shift, and drift of influenza. That's one of the topics up here. Okay? And then just being familiar enough with the terms that you can understand what I'm asking for. That's about it. Yeah. So any other questions from anyone? Sure. Go ahead, Ulrich. Oh, okay. Disease and infection. So like I was saying with the example of vaccination, right? So if you get exposed, or even not with vaccination, but if you have immunity, right? You can still, if somebody in your house, right, has the flu, you're still going to be breathing it in when they sneeze in your face, right? Or if you're taking care of a child that has the flu, you're maybe helping them to blow their nose, right? But if you have already immunity to that, even when the virus gets into your body, um, because of that really, let me share again, really quick response of your memory cells. Here, if we look at this graph again, you can see, go back, go back, go back. Here it is. Whoops, went too far. Okay, so in this graph to the right here, you can see that whereas at first, when we get sick with the flu or when we get our vaccine, we're having this kind of primary response. It takes time, and then when you actually get exposed to that seasonal flu that you had the vaccine for, um, then you won't get sick because it's so rapid and so much IgG that's present that your body will be able to prevent it from infecting enough cells to cause symptoms. Right? So you might breathe it in. It might get into your upper respiratory tract. Might even attach, and some of the virus might get engulfed by a host cell, and it might start to replicate. But because your B cells are immediately there and ready to make antibody to um, differentiate from the memory B cell into a plasma cell, which is an antibody factory, it just churns out lots of antibody. By the time the virus has finished replicating, there's going to be a lot of antibody present that will stop that virus once it's released from that host cell from infecting other host cells by that process of neutralizing the virus. So down here where you can get an idea, kind of picture of that here in this first example on the top picture to the right here, where the virus, once it's covered with antibodies, it can't get into another host cell, right? So that's infection, right? Getting into our body, getting even into a host cell. But disease is when there's damage, right? So we can have an infection, we can be even an asymptomatic carrier of a pathogen, but if we don't get sick, we don't get that damage that makes us have the symptoms, then we're not having the disease, right? Okay. I know it's often used interchangeably, which is why I want you to understand the difference between those two things. You're welcome. Anyone else have any questions? Okay. All right, good luck, and we'll see you later this week. And yep, and I'll end the session now. Okay, you too. Bye.